Hello!
Hi there.
Welcome to Microbiology Journal Club. We are now big about all things small.
My name is Danny, and in a previous life, I dropped out from my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker and editor for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC dedicating the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've also worked in bacteria, but I've also worked as a research integrity specialist. I'm currently working as an editor for a scientific journal.
Every week we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen during the week.
At the end, well, in the mid-month, hopefully we'll be looking into BioInk, which is going to be an exciting paper that we've been working on for some time. And please message us if you've got any papers that you're interested in. So one paper today was suggested by our viewers, so we're happy to dig into that. And so feel free, if you see anything that you want us to look at, please feel free to message at microtwjc.
Absolutely. We keep a record of all of the papers that we're looking at, either on the news weeks or during our deep dives in our shared Zotero library linked in the doobly-doo below.
Yeah, and we want to hear from you. So please use the comments or tweet us at microtwjc. And boy, do we have a show for you today. First up, we have two papers on SARS-CoV-2 therapies that could be put into nasal sprays. One targets TMPRSS2, the other is based on RNAi. Plus, we have a new technique for using blood glucose monitors to measure SARS-CoV-2. More on that later. And then we also have some articles on brand new phage therapies for acne and for vaginosis. Then we also look at how algae pick endosymbionts to partner with. And also we look at nano armor to protect probiotics. You can apparently armor bacteria now. Find out what that means later in the show. And also we have commensal bacteria that, again, betray us, picking up on thread from last week, how some commensal bacteria are associated with prostate cancer, and also how the smallpox vaccination can cause a bacterial infection, but that infection helps make it a better vaccine. What does that mean? Wow, that's all that in our show and more. So,
yeah. So to kick it off, we're starting with some SARS-CoV-2 papers. This one was suggested to us over Twitter, and it's from the end of March. It's titled "A TMPRSS2 Inhibitor Acts as a Pan-SARS-CoV-2 Prophylactic and Therapeutic."
Yeah, I think this is an interesting because we are having a lot of trouble with variants at the moment, which seem to be evolving in different ways. So there are there are some ways to target this. So there's some things that SARS-CoV-2 can't evolve away from. So thinking like the ACE2 binding, we've talked about a couple of therapies that bind ACE2. Because if it evolves away from binding ACE2, then it evolves away from being a better virus.
Right.
By the same aspect, the TMPRSS2, which is responsible for furin cleavage. For the spike protein, it works with two steps. First step, it attaches to ACE2. Second step, it needs to fuse with the membrane. In order to do that, it needs to expose its fusion peptide, which is this so it needs to be cut in half, and it uses host proteases like TMPRSS2 to do that. And so so this is kind of the killer app for SARS-CoV-2 that separated from SARS-CoV-1. It's what we generally attribute to why it's been such a horrible blight on our lives.
Yeah. But I think I was gonna say, like, the framing. I don't know if you remember, but last episode you were a little bit skeptical of this particular intervention, just because we learned that Omicron doesn't really require too much TMPRSS cleavage in order to get in.
Oh yeah, I was gonna save that elephant in the room for later, but
for later.
my j'accuse but we can talk about it a little bit now. But yeah, so I think the main thing that people need to remember when critiquing this paper is, let me scan down, because I think this is there's a very important detail here, because oh gosh, this paper was initially submitted a year ago on the 14th of May 2021. That is a full 5 months before Omicron appeared, and it was only accepted in March 2022. So when we look at this paper, we do kind of have to be a bit more generous when we think about it, because Omicron didn't exist when they were testing this or running these experiments. So they didn't know that it was possible for SARS-CoV-2 to evolve away from a dependence on TMPRSS2.
Yeah, because like you would think that it does, like, I guess before the point of seeing that particular variant, it was easy to think that they need to fuse, right, with
Yeah.
that fusion peptide. But I was looking through our Zotero library to find the paper that was talking about how Omicron doesn't use it. It's still a preprint. It doesn't seem like it was ever published, and it's Peacock et al. Talking about Omicron replicating in nasal epithelium, and then and they also sort of investigate the effect of camostat,
Yes.
which is the TMPRSS inhibitor, both on an ancestral strain as well as on Omicron itself, showing that it doesn't have as big of an effect on Omicron. So yeah, so we have that preprint against this this published paper. But I mean, the published paper is quite good in the sense that it seems very thorough in the way that it approached things, starting with checking things in cell culture and then moving into a mouse model, showing that if you give this, so actually, I think one of the big parts of this paper isn't even its the SARS-CoV-2 biology. It's really like this inhibitor, which seems like a really weird molecule.
No, I think I do want to talk a little bit about the molecule, because in order to do that, we have to talk a little bit about how proteases work, because proteases, they kind of have this, they're there to break things apart. So usually breaking proteins apart. In order to do that, there's a bit of chemistry involved. So in this case, TMPRSS2 is a serine protease. So it has a special like what we call a cassette of like 3 protease. So it uses a catalytic triad called serine. So it's serine, histidine, and asparagine. And serine is the thing that does a lot of the breaking, because it kind of has a moiety on it, a little part of the chemical structure that forms an intermediate with the protein it's attacking. So this is nucleophilic, I think is the term. So means it's like,
so the enzyme actually binds, if you will, right? Like it creates an intermolecular, it becomes one molecule with the thing that it's cleaving temporarily as part of the pathway to breaking that molecule apart. And it does that through this catalytic triad. They're amino acids that belong on the protein.
And so this nucleophilic, so there are 2 like types of like attack, I think, in chemistry. Remember, I'm going back to my high school chemistry here. So because there are nucleophiles and electrophiles, and that,
I was going to say, this is the tricky part of chemistry too. This is like Orgo, the most feared course in university curriculums for science majors.
I got a B in chemistry, and I'm still bitter about it. But I also, so that's how I know I'm bad at chemistry. But yeah, nucleophilic and electrophiles, they kind of, so nucleophiles, they're like attracted to nucleus, so they're very negatively charged. Electrophiles are very positively charged. They're attracted to. And this would be an obscure thing that, as a microbiologist, I never have to know. But in this paper, they talk a lot about creating these molecules that mimic proteins.
Peptidomimetic compounds.
And peptidomimetic compounds that have this thing called an electrophilic warhead, which is what a great term.
Seriously. So people are having a good time naming things. Scientists always have a good time naming things.
And the idea of this electrophilic warhead is when your protease comes along and goes, okay, I'm going to chop this other protein into small bits. It attacks this peptide, but the peptide has an electrophilic warhead that kind of binds to it and creates a covalent bond, and it kind of sticks in that active site and stops the protease from working. So.
Yeah. And so, that's the function of these molecules that they have developed and they're interested in testing. They're really quite more, at least 10 times more potent, right, than camostat, which is the inhibitor that almost every study we've looked at that's looking at TMPRSS2 cleavage is using that. This one, so that one's like at 17, right? The IC50 is at 17 nanomolars, and then all of these derivative peptidomimetics are looking at like under 5 nanomolars of affinity.
Yeah, and like a lot of this study is trying to make sure that these are like specific specific for the TMPRSS2 because we don't want because we do need proteases to live. So if we shut down all of them, it's not going to be good for us.
Yeah, yeah. So they create a very specific and strong inhibitor, and then they take it through the rigmarole of does it inhibit SARS-CoV-2 in cell culture? Does it inhibit SARS-CoV-2 in organoids?
Yeah,
right? They do
they use
organoids,
like
I think.
colon organoids, right? So looking at in different areas, and they kind of move. Yeah, they move gently up the scale from like different cell types until they eventually enter an animal model, which I think they use like a humanized mouse model.
Yep. Yeah, the K18 human ACE2.
Yeah, I think we talked a lot about that. We had an entire show on mouse models.
Definitely. Yeah, and so it works pretty well in the span of things there. And so, right, before knowing that some SARS-CoV-2, such as Omicron, don't exclusively rely on this pathway, one may think that this would be an amazing way to do it. But now, of course, like, okay, actually, looks like viruses do evolve away from this particular requirement as well. So maybe not as strong of a candidate as it was originally thought. But the technology of these very strong serine protease inhibitors, like that could be useful in other applications. Yeah, either therapeutically or even like in the lab or like for some sort of research goal.
Yeah, and I think another thing that the way they apply it, I'm not necessarily sold on this being the best, even if Omicron didn't exist. I'm not sure I'd be sold on this being the best antiviral, just simply because the way they tested in this study was essentially like giving it like quite frequently, like so you see this a lot in studies which test new drugs, or like they often like do a prophylactic route just to give the drug the best chance possible to show that it has an effect. And we've seen like that there are lots of drugs that do have great effects when you apply them like this, but when in a real world situation, it becomes a lot more difficult to see their efficacy. So
yeah, and it would also be like that taking a drug in that fashion also exposes you to more risk because you're being exposed to that drug like for a longer period of time. And then also if you forget it, then right, there are things that are sort of practically difficult with prophylaxis.
Yeah, and part of that is also like convincing people to take prophylaxis, because there is this argument that why should we have to take a drug for a disease that we don't know whether we're going to get? And you can only really sell that if it's something really scary. And I don't think people are necessarily as scared of SARS-CoV-2 as, say, HIV or malaria or other sorts of drugs were.
And certainly not with the presence of effective vaccines, right? Because knowing that the vaccines are reducing your likelihood of going into severe disease for most individuals, right? Not all, but like that wouldn't I think that logic sort of tells people like, oh, why would I want to take a drug then every day to prevent getting it? And in the paper they try to spin it, I think, on the side of like reducing transmission. But yeah, again, that just sounds like a very difficult sell.
Yeah, I mean, I've seen like this was associated with a couple of press releases that said it could be used in a nasal spray formulation, but that wasn't really tested in this at all. I think nasal sprays might be an easier sell because they're basically literally on the surface. So I think putting on a cream or a nasal spray is a lot easier to convince people. But the problem is like that isn't shown in this paper at all. So
Right. That's just some fun media speculation on the basis of papers. Yeah.
I mean, and of course, like all this is not taking into account that Omicron exists, which kind of like cuts this paper's kneecaps off, which is kind of unfortunate.
Yeah. But I mean, also, I guess it's like a nice time to just share that as well, right? Like we always think about like knowing stuff about evolution. It's quite surprising to see that Omicron, right, was able to enter cells through a slightly different pathway, right? relying more heavily on endosomal entry than furin-mediated cleavage, or yeah, furin-mediated cleavage fusion. And so yeah, it's kind of, maybe it's nice perspective, right, to also see that, right, this paper from just earlier in the year, how it really shook up the way people think about the virus.
Yeah, I think I've said everything I want to say about this paper.
Yeah. Yeah, let's move on to the next one, which I believe you also pitched at the very beginning as something that people say could be in a nasal spray.
Yeah, that's right. So this is a similar. This is a preprint. So again, it's not. So we'll probably have to wait a year until it gets into a real paper. But yeah, so this is a paper that sells more. So it's called Genome Widescreen of RNAi Molecules Against SARS-CoV-2 Creates a Broadly Potent Prophylaxis. So this is a paper about RNA interference. So what they did here was they took a panel of like the genome and tried to create. They screened over like 16,000 targets for RNA interference. So they did things like looking at what would actually get transcribed, what would actually bind to things, and and I think the key thing is that they want to sell here is they're using a new screening strategy called oh god, it's really like weird. I hate the name of this so much.
It's a specific name.
Yeah, it's called sense.ai, which is just impossible to Google, because the moment you Google at sense.ai, you get this like weird like you get lots of websites about like, oh, we can stimulate your brain to be smarter, and yeah, I wish. But
so it's a way of sorting through. They make like a huge amount of diversity of specific RNAs that they think will do some inhibition, and they have some clever method in which to figure out the ones that actually do inhibit.
Yeah, that's the long and short of it. And so they run this, and they do like another version of the screening assay using the same genome but using older techniques. And then based on that, they took like the top like candidates from both, and they checked to see whether they worked. And they found that the candidates they found through this sensor AI technique tended to be better at inhibiting SARS-CoV-2 than the other candidates. So this paper isn't just about like selling this as a treatment, but selling this kind of workflow for finding interfering RNAs.
Right. As we've seen in a lot of SARS-CoV-2 papers, right? There's like core understanding SARS-CoV-2 papers, and then there's sort of papers that are saying, look at these other techniques that we use and how it's applicable to SARS-CoV-2.
Yeah, and so this is like an SI strategy. So the idea is that this will be a formulation that will get. So again, they're a bit more explicit here that they want to use it in a nasal spray, and then this nasal spray will have this like small interfering RNA that will enter into the cells that are infected with SARS-CoV-2, and then bind to SARS-CoV-2, the SARS-CoV-2 genome, and like alert the host about things and start using the host like kind of genome to chop up SARS-CoV-2 and stop it from spreading.
Yeah, the system. So we have like, I think all vertebrates have this system. I think the system, there's also like a form of the system that exists inside plants, where I think it was first identified. Essentially, there's a certain signal that the system recognizes in RNA that then chops up that piece of RNA and uses the resulting pieces to direct RNA degradation. I mean, it's very CRISPR style, actually. This system was learned, we knew about the system like way before CRISPR as well. And yeah, it's a set of proteins that does this. And they're able to stimulate it with sequences that are specific for SARS-CoV-2, and thus demonstrate in cell culture that it works.
Yep. And and in cell culture they decided to not just look at one at a time, but also look at cocktails of different things. So in like figure D on here, they take their best activated, like the RNA that works best on its own, and then test it against these paired cocktails to see which ones were better. And then they took those formulations into Syrian hamsters, which we know are a pretty good model for SARS-CoV-2 infection. And again, like they found that they did better, but again, pretty much every single critique the only critique that we don't have about this, because they did test on Omicron and they found it worked on the Omicron strain. That's because they had the that available to
Yeah.
them. So, but apart from that, pretty much all the other critiques we had of the previous paper apply to this one.
Yeah.
That it's still like a very early stage RNA. The way they deliver it is they do give like almost daily doses of it. But then again, they're trying to like say that maybe if we did it in a nasal spray, it'd be a lot more convenient for people. So
and then maybe
yeah,
just the platform of being able to screen through different RNAis like is something that people will use in the future, like not SARS-CoV-2 related, right? But for other
I think that's
applications.
the main thing that will probably be lasting from this paper.
Right. Okay. And our final SARS-CoV-2 paper today is about a diagnostic tool, a simple and universal nucleic acid assay platform based on personal glucose meter using SARS-CoV-2 N gene as the model. So yeah, I was interested in this just because we've talked about interesting SARS-CoV-2 diagnostics. I mean, we have lateral flow tests, we know, in abundance, looking for protein antigen, but there's still interest. I mean, at the end of the day, PCR is like one of these, it can find it a little bit sooner, or nucleic acid-based techniques might be able to find infection a little bit sooner. And of course, a big concern is making sure that assays are like really easy to perform and adapt, right, to different scenarios. So again, maybe this is just about the technology more than its application to SARS-CoV-2, especially because as I was reading through this paper, they don't actually ever use like any clinical samples or SARS-CoV-2 itself in it. They just have like a model. They have like a piece of DNA essentially that matches a SARS-CoV-2 gene, and they use that as their SARS-CoV-2 test.
I mean, yeah, they do a lot of like interesting. So the basic idea is like they take a glucose meter, the stuff that diabetics we would use to monitor like glucose, and you kind of can use this test to like convert have like an enzyme that detects SARS-CoV-2 and then converts something into glucose that can be detected by the glucose test.
Absolutely. Like from the detection point of view, they've done a lot to make it a very accessible test. So I don't know if folks remember, we did a deep dive on a CRISPR technique that used a cell phone to look for SARS-CoV-2. And you know, a lot of what I was excited about when we covered that paper was the accessibility of that technology, right? Oh, cell phones, right? Something that you can, a CRISPR protein you could express, right, inside of some small system. And here essentially it's a type of sandwich ELISA that they're doing on a magnetic bead, where they take magnetic beads that have like a sequence that's fishing for the SARS-CoV-2 sequence. If it finds that sequence and it grabs onto it, you can then remove everything else in the sample. The authors do a lot talking about the advantages of this particular part of the procedure, where it means that it's sample agnostic. Because anything that might interfere with the other parts of the reaction, that can all be washed away at this step. So like stuff in your saliva or like other contaminants, right? Only the things that are binding to those magnetic beads is going to move on into the next stages. And so it removes some of the interference potentially of later steps. And then what they do is they have the other part of the sandwich, the top of the sandwich, which also recognizes that part of SARS-CoV-2, and this one is attached to invertase, which is an enzyme that breaks down sucrose into glucose and fructose. And so that's what ends up, and so their detection is based on, they add a bit of sugar into it then, and they say the amount of invertase is proportional to the amount of target that they've pulled down, like SARS-CoV-2 target, and so thus the amount of glucose that gets measured by just these diabetes glucose meters is what is used, is proportional to the amount of sample being pulled down.
I mean, yeah, it's very interesting. I mean, from a DIY biology perspective, it sounds like you got all these things. I mean, we just heard in the comments that even keto people have these. So this is going to be like,
The device is everywhere.
Everywhere. My only like concern with it is that it does seem like there are a lot of like steps and a lot of reagents required, and they did. I mean, they did some cool stuff to like try and minimize background. So they tested it. So they put their DNA samples mixed in with salmon sperm to try and simulate like having lots of off-target like
Yeah.
But yeah, I think they said that it takes about 2 hours to incubate in order to get an accurate result, and they didn't really do much like benchmarking against, say, the RNA tests or the kind of PCR tests, which we would like kind of hope that this would be a substitute for. So this doesn't seem like a full demonstration that this technology is going to be like the next lateral flow test. I mean, it seems like.
I mean, I think again, like they don't use RNA either, right? Like they're using, I think, DNA DNA things that match. So like that presumes that people are doing like an RT step, but that's like a complicated thing that is hard to work into workflows.
Yeah.
So
it's an interesting concept, but I don't think this paper's really sold me on it.
Yeah, same. but I want to pick it out because I'm interested in like just these other ways of measuring. And it seems like there are other people have been working on this technique. And I think that there is real value in figuring out like diagnostic techniques that aren't just like behind sort of more complicated methodologies, and having those platforms ready to go for the next time, or even for like the next variant potentially, like that could be really helpful.
Yeah. I mean, I like the idea of using these glucose tests as almost a platform for doing science at home. I like the idea that you can like order, if you have one of these tests, you can order a kit to measure other like biomarkers potentially. That's kind of a really fascinating application of this.
Sure. I mean, it's like an easier ELISA. Well, I'm not sure if it's an easier ELISA. I was reading the protocol. They do their own like invertase conjugation to the other part, the top part of the sandwich. And I actually am not really sure what like buying those reagents is like, or doing that particular chemistry.
Yeah, I mean, I like the idea of it. I just don't think necessarily it's best. It's not showcased very well in this study.
Yeah.
But I like the idea of it, and I think the concept's very interesting.
Absolutely.
Anything that lowers the costs of science is just something that I find quite interesting.
Yeah. Yeah. Important, I think. Very important. that's why I do what I do at Biotech Without Borders.
Yeah.
Okay. Let's move on. Yeah, let's move on. So we got towards phage therapy. Oh, so we're moving out of SARS-CoV-2 into the wide world of microbiology, which is very large. And this one, we're looking at phage therapy for acne vulgaris, topical application in a mouse model. So I didn't even know there was a mouse model of acne, which is also, it's a weird model. They inject intradermally this clinical isolate of acne, and then they cover the mouse in human sebum, and that creates the inflammatory lesion.
Yeah, it's a very like strange model, but I can sort of see how it's like it's very hard to simulate, like, because again, the injection isn't quite the same as a pore, but yeah, the idea they want to like simulate a human acne infection so they can test whether this cream they create, which has phage in it, can make the lesion smaller. It's not often I see like phage therapy papers around. And so what they did was that they isolated phage. So there's like this culture collection that they created a while ago, and they picked out some phages from that as like phages that are for this Cutibacteria acnes, formerly known as Propionibacterium acnes. So just in case there are any of those fans in the audience. And the thing they were keying in on is a lot of acne strains are now becoming antibiotic resistant. So they thought, well, let's test whether some of them are susceptible to phages. And they found they tested 36 strains, and 11 were resistant to antibiotic, and I think only about about 4 were resistant to phages. So there is still a high degree of this background phage resistance, but it's still better than antibiotics. And what they kind of found was that the phage resistant isolates.
I was going to say, and phages are more targeted than antibiotics, which is like a nice way, right? We're not just like covering ourselves in antibiotics and hoping we kill the thing that we want, but also killing a whole bunch of other things. Phages are a fairly targeted therapeutic. Mm-hmm.
That's a very important thing to note, because sometimes you don't want to get rid of the whole skin microbiome, lead to worse things. So having like a targeted assassin to kill off the one thing that's causing the problem, that can be very attractive. And the thing I want to point out is that the antibiotic-resistant bacteria were never phage resistant, and so the idea, like, maybe you could combine these somehow, but then again, that kind of screws up the point before of like, we don't want to kill all the bacteria. But yeah, so they go about. I mean, it's an interesting concept and an interesting model. They go about it in a rather strange way. So they do create some cream with phage on them, and they measure the how well it's working by taking photos and then seeing how like much smaller the lesions get. They also like do things like how raised they are. So just thinking about, like, we've all had pimples, I assume. So there's like pimples can like be wide, and there's like a bump, so you can try and like get some volumetric analysis to try and think, okay, how raised is that bump? How wide is that? But I mean, measuring that can be a bit. There is a bit of, because if you measure with calipers, if you squeeze the calipers too hard, maybe you can make it look smaller than it is. But it's so with this, they take photos, so it's a lot easier to be standardized. They don't actually take
figure four.
Yeah, figure 4 has the pictures.
I mean, it's definitely not a pretty sight, but yeah, those are what mouse pimples made of human bacteria and human sema on the back of a mouse look like.
Yeah, they shaped the mouse's backs and they put it intradermally, and then they measured like the diameter score and elevation score, and they found that with phage, when phage was in the cream, they were lower. So that the phages were doing their job there, and they were specifically looking at lytic phages, the phages that pop open bacteria and kill them, rather than the ones that just sit in the genome and are really selfish about it, which we usually talk about.
And they're able to then like section these, right? They can look at the immune response, because this is an animal model. They get a lot of data. You get a lot of data out by harvesting the lesions and figuring out what's going on. And so the phages do reduce the immune response that's going on, which I mean, like, immune response is partially why we get these lesions in the first place, right? So it's sort of like phages are destroying the bacteria so the immune system doesn't have to, I guess, is part of the story.
Yes, so they were doing like daily application of it for like 5 consecutive days, and they found that there was some broad improvement. I would have really liked to have seen some more like actual colony forming units, so you can see like first at the end of it whether there are any resistant bacteria, and also like whether the whole numbers were being decreased as well, or whether it was just modulating the population. But aside from that, it's quite interesting.
Yeah, I find it a weird model, I guess. I'm very fixated on the injection over topical application. I mean, I think that was like part of what I was thinking about a lot in grad school. Like, I was putting bacteria in the top of the skin because like there's like abscess models where you inject it, right? Or like there's skin lesion models you inject it under, but like you're bypassing all these other interactions that happen on the surface of the skin. But I guess one thing you could say about this is that I mean, I don't know too much about acne, but I think it does start from like the hair follicles or whatever, and that's like underneath the layer of the skin. So maybe that's a bit better.
Yeah, I guess it's kind of like ensuring that, because if you get deep in the skin, there is a skeptical like, do the phages actually get down there? Is it possible for? So I think that if they could demonstrate,
they're addressing that part. Yeah, the phages somehow are getting lower, so that's
making it past at least like 2 layers of integument of skin, and that's quite impressive.
Totally.
Oh yeah, I mean, there are also some. Do you ever work with scarification models? Because they're the worst.
No.
Sounds
It sounds horrible for the animal.
It is horrible for the animal and horribly dangerous for the person. So I think
Oh,
there was
really?
like one example of someone giving themselves smallpox from it. Because
Yikes.
yeah,
No,
it's
because like the same thing that you could scratch the animal with, you could scratch yourself with, right?
yeah,
Is the idea. Yeah.
yeah. And scientists aren't necessarily known for our sportsman like agility and reflexes, so sometimes accidents can happen in the lab. I mean,
Uh-huh.
so yeah, so for those models, it requires a high degree of coordination and kind of skill. Um,
Right.
but yeah,
Okay, well, let's I think we can move on.
let's talk about this. So, identification and characterization of novel endolysins targeting Gardnerella vaginalis biofilms to treat bacterial vaginosis. Now, this is interesting because because traditional phage therapy, you add the phages, whereas this is just taking the aspects of lytic phages that causes the bacteria to die.
Mm-hmm.
Which so
The
endolysins are,
endolysins. Yeah.
yes, they're fantastic little proteins. And I like this as a strategy because with normal phages, there are lots of bacterial defenses against them. So there's like DNases and there's CRISPRs, there's
Mm-hmm.
all sorts of things. But none of those, but there doesn't seem to be a focus on bacterial defenses against specific proteins. So
Mm-hmm.
so the thought I was having was that maybe that and go to turning endolysins into something druggable, that could be an interesting therapy. The only problem, yeah,
Yeah. I mean, I was going to say that this is, I think, actually is an emerging line of thought. When I was in grad school, people were looking at endolysins against Staph aureus as a potential way. And from what I've heard, that research continues just because there's so much diversity within the phages, right? Like there's a lot of different endolysins and you just can look through all of them essentially to try to find the best one for your particular target bacteria.
yeah. And I think going back to the original point, that endolysins are also very specific to some bacteria. So they're
Mm-hmm.
probably not going to be as broad spectrum as antibiotics. But if you want to assassinate a specific bacteria in a particular gene, a particular microbiome, then this would be the perfect tool to do that. And
Yep.
that's
Yep.
what brings us
And
to, sorry.
oh, I just want to say, and so for this paper, they're looking at this particular bacteria, Gardnerella vaginalis,
Yeah.
and similar to actually a paper that we were looking at last week, I think, right, with a gut microbiome member that sort of sets up the biofilm for things. I think this bacteria has been identified as an early colonizer of the vaginal
Yeah.
surface in bacterial vaginosis, and its ability to make a sticky matrix to grow in, uh, uh, essentially gives some resistance to any other bacteria that then comes along, can sort of co-colonize that and drive the pathology of that particular
Yeah,
disease.
exactly. I mean, it's like the key like kind of planner of the bacterial vaginosis infection, where this is
Uh-huh.
like the first bit that makes all. It doesn't necessarily do the worst part, but it makes it easier for all the other members of its team to do the worst part.
Right,
Um,
right. City
so they,
planner.
yeah, it's kind of like
Dysfunctional city planner.
yeah, city planner. Or I guess if we want to go with a more war metaphor, I guess like the general or the strategist, perhaps. Um,
Sure.
But yeah, and so the idea is like, wait, let's take an endolysin that targets this one specifically, and they found like that this is particularly vulnerable to endolysins because it's got like exposed peptidoglycan,
Mm.
and the endolysins that work on it are ones that break down that cell wall.
Hmm.
So this so they devised a couple of endolysin candidates. They decided to test against it.
Yeah.
Um,
Yeah, I thought it was interesting the way that they fish through the genome of this species,
yes,
actually. Just looking at like multiple iterations of the species, looking for the phages that are integrated in it, and just picking out the endolysins, being like, okay, well, this is like already associated with this particular bacteria. What happens if we kick it into overdrive, make a bunch of it, and see what happens?
yeah, it's a really interesting thing because, like, again, it makes because fishing for phages is almost a lot easier for fishing for antibiotics because all you have to do is look at the bacteria that you're targeting and then pull out endolysins and then doing a little bit of engineering on them to try and to improve
Hmm.
their like maybe specificity or activity, and then you've
Hmm.
got something that you can use. Um,
Absolutely.
So like they tested this in biofilm models, and I think they used baseline, and they found that most of them were like almost as good, I think, as the antibiotics. Maybe not necessarily better, but they're not looking for better. They're looking for something that kind of an additional thing to use in their toolkit. And so they tested them in various biofilm disruption assays, and they found that they're not like groundbreaking, but I think for this sort of thing, they are looking to assassinate a specific area of the microbiome rather than doing a
well, in table 4 they show that it doesn't work against these lactobacilli,
which is good because lactobacilli,
which is really good,
are our friends. Yeah, I think that's the main thing. Like, we want to kill off the bad bacteria without hitting the good bacteria, and this study kind of demonstrates how endolysins can be a good tool for that.
Totally. Okay, I'm good with that. Let's go on to our next one.
Yeah. Oh, this is an interesting one. Symbiont. Do you want to read the title?
No,
okay.
Go ahead.
Symbiont-induced phagosome changes rather than extracellular discrimination contribute to the formation of social amoeba farming symbiosis.
I remember reading the paper that was talking about the identification of the social amoeba farming symbiosis, which in the title of that paper, they're careful. It's so tricky, right? Like, to talk about amoebas doing farming. It's like a complicated behavior, and like, what exactly is that we're studying? But the long and short of this phenotype is some amoeba will hold a specific species of bacteria inside of themselves, and then so during starvation times, when they form these like spore forming structures, I think this is if you remember, maybe was it 2 weeks ago now, we were looking at the hyphae amoeba, the ones that had the follower things, and when they got starved, they become like spore structures to disperse themselves. And so these amoeba, this is one of those types of amoeba, and they take in a certain bacteria into their bodies. So then when they go into the spore structure and they disperse themselves, they seed their environment with the bacteria that they held within them. And that was called farming, because they're essentially taking their food with them and going to a new environment, and then I guess spitting it out. Maybe some of them die, I guess, and then those bacteria grow, giving food for the rest of the amoeba that are in that area. And so that was like an interesting phenotype that was discovered. It doesn't happen with all of these social amoeba, so that's an important observation from the previous papers that described the phenotype. Only some, and so I guess choosing then a farming amoeba and an organism that does that goes into them, that's a model system to study this particular behavior. And what they want to do in this paper is figure out whether or not the amoeba are like preferentially just finding these organisms in the environment, or maybe discriminating them in a different way. And that other way, which they find is the case, is intracellular discrimination. So something about once the bacteria go into the phagosome, then is then the amoeba does some sort of sensing. But before that point, the amoeba don't have any preference in terms of grazing on symbiotic or non-symbiotic types.
Yeah, so they eat everything and let the endosomes sort it out, basically.
Yep. Yep, absolutely.
Which is a fun way to think about it, because you got these two because Burkholderia, there's like the symbiotic version, and then there's one that isn't. And then I think that tells us a bit about like how actually like it's not like a passive process. The bacteria that are in symbiosis have to do something to make sure that the phagosomes are like okay for them to survive in.
Yeah, so absolutely. I mean, that's actually that's so very fascinating. I mean, we've read a bunch of things now, I think, along these lines of like how do you set up endosymbiosis, or how do you set up a symbiotic relationship? And there is something where you got to inject things sometimes into what you want to make symbiotic with you, because the things that you inject, the way that you modify your host, there's a way to do that that's pathogenic, and there's a way to do that that says I'm friendly and keep me around.
Yeah, like symbiotic relationships. So both partners have to do something. You can't just. It's very hard to have just one partner who just does all the work, and that's not a great relationship. That's like almost a parasitism. So this one kind of indicates that that both these partners in this relationship are doing something to make sure they can stay together.
Yeah, I think that's what they find here. It's a pretty that's what they got. They have the two assays, the pair choice assay, which is what do you graze on, and then they go in and they look at the survival essentially. Oh, and the pH levels. They look at the pH levels inside of the phagosomes, and they find that in the symbiotic form, those pHs, they don't go as low. So they're not going down the digestion pathway. They're going down some other pathway of co-living.
So that.
The one thing I want to say about this is that I'm interested to know, like I sort of said, like, oh, like maybe these bacteria, like I talked as if I knew certainly that these bacteria are injecting something into the amoeba. That's just like a pattern that we see a lot in the way that bacteria live in phagosomes, is that they have a secretion system where they inject something into the host that modifies the way that phagosome is done. But I mean, that modification, like it could come both ways, right? It could also be sensors inside of the phagosome that are looking for specific molecules, right? That then direct the fate of that phagosome to something different.
Yeah, that's the interesting, because we don't know whether there's just one single pathway to endosymbiosis. There could be lots of different ways. I mean, there are different ways that various pathogenic bacteria can manipulate like phagosomes. I mean, we've seen it with listeria and tuberculosis, where like, or any organism in the environment that gets eaten by an amoeba, they've gotten
Legionella. Legionella is like the classic eaten by an amoeba bacteria.
Exactly, and those sorts of things can be pathogenic for humans, but it may be in the amoeba situation it's different. Things that get complicated.
Well, there's also a lot of. I think the other thing is, in those pathogenic forms, like we're focusing on the pathogenesis, right? But like there's also space within science to, you can ask whatever question you want in science, and if you're being rigorous and you're creating the right methods, like you'll find some insight into it. So maybe like asking questions about like, right, the breadth of diversity inside of protists and the breadth of diversity inside of their potential pathogens, finding those overlaps, but then asking which ones might lead to symbiosis rather than to pathogenesis might give us like insight into the symbiotic pathways rather than just the pathogenic ones.
And that brings us to our next paper. It doesn't really bring us to the next paper, but I need to do a segue somehow. A single cell nanocoating of probiotics for enhanced amelioration of antibiotic-associated diarrhea.
This is so crazy. This is the bacterial armor that we were talking about at the beginning.
Yeah. Because
go ahead.
I'll let you take it away on this one.
Okay. So essentially, we know that antibiotics destroy the microbiota of our guts, and that could lead to bad outcomes. So maybe there's a thought that you seed it with some of your favorite probiotics, and that will prevent your gut from experiencing those bad side effects. But there's still going to be lingering antibiotics in your gut if you've just had antibiotic treatments. So how do you make sure that the bacteria, the probiotics that you eat afterwards, aren't just being killed immediately by those lingering that lingering antibiotics? Or maybe how could you propose a way of delivering both your probiotic and your antibiotic at the same time, so that you can kind of do a reset, essentially? And the technology that these guys, that the authors of this paper, I guess, are describing that could achieve this is some sort of phenolic it's phenolic, right? A phenolic compound that coats a single bacteria that essentially absorbs antibiotics, preventing them from getting into the bacterial cell, so that then when everything's calmed down and the bacteria decides to divide, it just, it sheds its armor and becomes able to grow.
Yeah, it's a really neat sort of idea of, I mean, it's not like they call it nano armor, which is such a cool like idea. And yeah, the great thing about it is it's supposed to like not produce antibiotic resistance, because you're not adding a resistance gene, making this coating that when the cell divides, it like the coating like kind of dissipates. And I think it does get carried down through generations, but eventually it gets to a point where it isn't protective at all. Which is really good for making sure that antibiotic resistance doesn't enter into like the rest of the gut community.
Yeah, so that's what they do here, and then they test it out in a rat model of antibiotic induced diarrhea. And so these rats, they can be given levofloxacin, I think is the antibiotic that they use. And you can either get regular antibiotics co-administered or your armored probiotics co-administered, and then just checking out the whether or not those bacteria show up in the poop, and also the quality of the poop. So there's also wonderful pictures of rat poop.
Yeah, you get these like, this Bristol stool chart, which is like the alternative personality test I use. What score are you on the Bristol stool chart? So yeah, really interesting like concept. I like the idea, because the thing about these, it's not like this armor is like bouncing off the antibiotics. What happens is the armor seems to be like binding and inactivating the various antibiotics, and that means that all the other nutrients that the bacteria needs to survive can still filter through. And
actually, I think they investigate closely a little close in Figure 3. They look at how that armor works. Right? They use essentially they're just looking at what absorbs to the armor. They coat like a chip with the armor instead, and see like they're able to see like how much antibiotics stick to the surface. And all of these different antibiotics, Cipro, tobramycin, neomycin, levofloxacin, norafloxacin, gentamicin, they all stick to the armor. And so it's anticipated that it will be effective against all of these.
But this is a really interesting technique. And I don't think I've got much else.
It actually makes me think of like candy. I don't know. There's something about like, the process of coating candy, like with a sugar coating. I'm just like, now our probiotics are coated in like a little coating that allows it to survive antibiotics. Very interesting.
I mean, they even like have little diagrams of like the pill you put the probiotics in. So there's like several layers.
They have their gastric protection, and then they have their intestinal antibiotic protection. And they and they're a different color too. In Figure 4, you can even see the armor, I guess. Like it darkens the cells. I thought that was kind of funny.
Yeah, that is. Oh yeah, I see.
Yeah. Okay, let's move on.
Move on. So this one is following on from last week, where we did talk about how sometimes in certain situations your commensal bacteria can betray you. This paper is looking at the microbiomes of urine and the prostate. Microbiomes of urine and prostate are linked to human prostate cancer risk groups. So this is kind of a correlation study that looks at people with prostate cancer and their microbiome to figure out what aspects of the microbiome community are associated with prostate cancer. And then
so they might not be betraying you. They may just be like correlated with your cancer.
This paper is a lot less functional. So the last paper, they actually tried to they did some experiments to see the bacteria disseminate into different tumors and help with metastases. This looks very much like at the broad in like the human situation. The last paper was mostly focused on mice cystox in people and in prostate cancer specifically. Look, and so it like
yeah, like a real
they do things like
clinical association here is what they're uncovering.
Yeah, so they do things like take patient urine samples, and they also take like cancers, and I think the term they use is they squeeze a prostate tumor to get some of these things out, which is
that's how science works.
We're adding to the great images. To anybody who's eating while watching us right now, you're welcome. So yeah, I mean, this paper they do focus bacterial candidates who are different from the ones we saw from paper last week. So, and they also looked at anaerobes. So a big part of this paper is trying to sequence uncultured microbes, because that's a big part of the microbiome. And so their thought was we need to make sure we have look at these anaerobic bacteria. They might have an effect on it.
And they find the associations.
Yeah, they find the associations, and it's a bit woolly because obviously it's correlation, not necessarily causation here. I mean, we've got prior evidence that maybe make us think it's causation. They also looked at the genome to see if there are any shared factors that could give a clue on what the cause, whether there's like a specific pathway that these bacteria use to cause cancer. And they basically couldn't prove anything conclusive, which, based on the previous study, is expected, because the previous studies kind of said that these bacteria work by tickling the immune system in a specific way, and there are lots of ways that bacteria can activate the immune system. So it doesn't have to be a single pathway that links all these bacteria together. But yeah, I thought that well, basically I saw this. So after last week's show, I saw this in a press release. So I was thinking, gosh, I should have talked about this.
Instead, or just in addition to?
Just in addition, not instead. Yeah. I mean, again, I think last week I spent quite a long time talking about that paper, so it might be a personal obsession of mine.
And I do agree with in the comments. Like, I think that the authors also are trying to form this more as a biomarker study, right? Rather than understanding things. But again, having an association found in human samples is also a very good data set to springboard, right, and start asking some functional questions. But as it stands now, the information that they gave us in this paper, much more along the lines of diagnostics. And maybe it would help, help confirm other testing methodologies, because I hope people are also aware that in radiology, when you look at radiological data looking for cancers, like that too is not a perfect science, and that there is the possibility of false positives and false negatives. So having another way of having some sort of biomarker to suggest that cancer could be there, maybe direct a biopsy, or gain confidence in a specific treatment modality, that's very useful.
Yeah, and I'm all for any early diagnosis of prostate cancer is such a good thing to have. Because there are certain ways to test for prostate cancer that are embarrassing to talk about at the moment, but are very important to do.
I was gonna say also, I think they're gold standards for a reason, right? That's interesting. Thinking about wait and watch, and I think this is definitely territory that I'm not super comfortable in speaking in, but there is sort of a way in which doctors approach, right, and having more information can help them differentiate and potentially act faster than they normally would have. Okay.
And the next paper is smallpox vaccination induces a substantial increase in commensal skin bacteria that promote pathology and enhance immunity. So this is an interesting one. So they're looking at vaccinia virus. So this is the virus that was that we think emerged from cows or horses. There's a lot of mystery over where the vaccinia virus actually came from, apparently. But it was used as a smallpox vaccine.
That's the classic variolation, right? The style.
So they look at, so I think in previous work they looked at a mouse intradermal vaccination model, and and they found that there were like lots of neutrophils and T cells, which is kind of very different from how like it responds to how respiratory infection would go with the same model. So the question is, what is different on the skin and in the lungs? And so the idea they thought was maybe there's something in the skin microbiota that's causing this difference. So what they did was they tested. The first thing they looked at vaccinated animals, and they found that there are some differences in the skin microbiota. There's some inflammation happening. And so the question is this due to the microbiome or is it due to the vaccination? And so what they did was they do a lot of tests with antibiotic treated or germ-free mice as controls to see like what effect the skin microbiota is having in this effect.
And the lesions that they get in the specific pathogen-free are a lot bigger, right, than the lesions in the germ-free. And again, that's like usually skin lesions. A lot of like visual aspects of a disease are being driven by some inflammatory response from the immune system. So it does indicate that we need those, or in this particular model, those microbes are required to create this type of this inflammatory response.
Yeah, I mean, we talk a lot about adjuvants and how important they are, but what seems to be here is that when you get injected with the vaccinia virus does some things to the immune system that make conditions like better for the microbiota to become infectious effectively. So yeah, this is still carrying on the theme of your commensals. Given the opportunity, we'll try to eat you alive. So. And in this case, the vaccinia virus gives.
But also in this, right? But then in this one, they're trying to as well say that that immune response is the immune response we want.
Yeah.
Like we do want to have this type of like inflammatory style immune response in order to make good antibodies against the thing that we just put into somebody's body. So this inflammatory thing, it's like, it's making a worse lesion, but it's also driving the actual function of the vaccine.
Yeah. So what they find is that when this bacterial kind of like secondary infection happens, it allows the mice have a better immune response to vaccinia. So this is like the third act twist where the bacteria weren't betraying us the whole time. They were like Jack Sparrowing us, where they're actually on our side the whole time, but they made it look like. So it was like, quite an exciting kind of turnaround. This is the contradictions.
In figure 7 is that turnaround, because they're just getting better vaccinia specific antibodies in the non-germ free model.
So in this sort of case, it's almost like the microbiota are being co-opted to act as like an adjuvant in this case to make the immune response and inflammation worse, which kind of looks bad, but also in the final outcome makes things slightly better. So
it's actually this is kind of hilarious because I've been listening to a song by the Tragically Hip called "Vaccination Scar," and then I was reading this, and it's like there is this like focus on like that scar of vaccination, and saying like oh it's so bad, like you could imagine someone framing it as like, oh my God, like you got infected there. That's not a good thing, right? But then actually, like maybe that is the thing. Like that's part of how this vaccination works. And better that than getting smallpox, right?
Yeah. And it might even reflect something about smallpox as well. Because again, like it's fortunately, we hope extinct, but it might reflect that bacteria microbiota might have played a role in smallpox infections. So
sure. Yeah, absolutely. Yeah. And I mean that I think we touched a while back ago about like mouse models for smallpox infection itself. And I don't expect that we'll see a lot of research along the lines of figuring out how smallpox works per se, because again, it's more about like control and so forth. But that is actually really interesting that it might be this observation could play into understanding the pathogenesis of smallpox as well.
Yeah, and that's all I have to say about this preprint.
Cool. So for our last paper, just a hilarious title, Thor's Hammer, the antibiotic corysine drives gene expression in a model microbial community.
It gets even funny when you see what THOR stands for.
Yeah, that's right. Right. Thor is oh man, hitchhikers.
The hitchhikers of the rhizosphere.
So yeah, I guess it's a super group essentially.
Yeah.
A
Maybe we
super
should
group
say
of
that as well.
hitchhikers. So it's specifically three bacteria that I suppose they observed. This group seems like they've studied these hitchhikers as a team because the keystone of it is this Bacillus cereus that has been shown to improve growth of plants when you spray it over a garden or a certain plant growth. But how they found the other hitchhikers were they found that, oh, it actually seems to always associate with some common partners. And so they extracted these three members of the community in order to study them in isolation. And previously, the authors have found very interesting interactions. Like the pairwise interactions of these two things are not the same when you add the third one in. So there's the way the signaling is happening in this set of three. They're highly dependent on each other. And in this paper, they do RNA-seq essentially to figure out like, let's get a big data set on how those things are different in the different pairwise interactions. And yeah, they are very different. I think the good way to see the data, or the important data, I think, is in figure 2. Figure 2, they have three in figure A, B, and C. These are differentially expressed genes. And so, like, right, you can be differentially expressed, meaning that you express less in a certain condition, or you can be differentially expressed as you express more in a certain condition. And then the two axes on this graph are against each of the members. So first we're looking at just this F. johnsii against B. cereus, right? Does it express more or less? And then the other one is F. johnsii against P. carensis. Does it express more or less? So typically you'd see this as, if there was only a single axis, this would be a volcano plot. But because there are two axes, right, two different expression, I guess, interventions that you could see, then it looks like just a mess. But each of the four quadrants represents either plus or minus, right, against one of these, one of the pairwise interactions. And so, yeah, they have different interactions depending on what they come together with. And they show that that's mediated by one of the antibiotics that gets made by, I think, the Pseudomonas makes a specific antibiotic, and that's governing some of these changes, but it also keeps the growth of one of the members, not the cereus, the other one, the johnsii.
Johnsii, yeah.
It keeps that growth in check, so you don't get too much of that, and it sort of balances it all out. And I find that very reminiscent to, again, I guess today I'm just thinking about how all of our literature connects to other literature, but like I find that very reminiscent to the synthetic bacterial communities that were being made, where they engineered, right, ways of each bacteria controlling each other based on central metabolism. But here, actually, in nature, no one would ever become that dependent, I think, on each other, because they're still free-living organisms that sort of need to do their own thing. But there are still those types of very close relationships achieved through, well, in this case, they're highlighting antibiotic production.
Yeah, I mean, this kind of like also calls back to things the vaginosis. Again, that does form an evil super team, whereas this one is kind of forming like it's kind of interesting because I've been reading into this, and like the things like the Bacterocereus releases fragments that allow F. johnsonii to grow in the root, and then Corensis produces an antibiotic that kills off the johnsonii, but B. cereus kind of like can protect it. So we've got this like triumvirate of like these relationships where they are all dependent on each other in this environment, and it's this paper just digs into that a lot more. And
Yeah, absolutely. I mean, it gives this now I guess this, so again, this research group has been looking at these interactions between these three for some time. So, but now they have like a genetic map, right? That they can start, it can drive their hypotheses. It could try to explain some of the phenomena that they've already seen. Yeah, it's just like a really good tool. And again, these model communities to study, I think, are very useful because we want to make our own communities, or we want to understand how to interact with these bacterial communities that live on us and in us and around us. And so like understanding some of the ways in which those interactions happen, yeah, I think is just a really useful piece of information and just fascinating, right? Like in terms of the diversity of communications that happen in the microbial world.
Yeah. So I guess like another thing I'm thinking of is like, this is quite like an esoteric paper, but it's just like, I'm so surprised I haven't seen a press release saying Thor of the Avengers, this other team up of bacteria. Because
Yeah.
that is a kind of obnoxious.
Well, I think the authors want that too.
Oh yeah, definitely.
Yeah.
I mean, like one of our commenters said, the niche of the niche, the nerdier the title.
Exactly.
I mean, they
exactly.
get like various things like named after pop stars. I remember like I once tried to get a job at this place called the Brad Pitt Consortium, which is a really tortured acronym just to get the name Brad Pitt into it, which is just
Yeah. So that's all we have for you this week. Join us next week for more cool microbiology news.
Yeah, thank you. And while we are like enthusiastic about microbiology and somewhat qualified, it's possible that we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter at microtwjc. Of course, in the comments during our live sessions. We both believe that peer review is a process that we can all participate in, so we hope you have a good time listening to us ramble on about microbiology today. If you think you have something to add or found something unclear, just let us know.
Yeah, it's been a pleasure chatting to you, Danny, and it's been a pleasure like reading the comments of Andrea and Minref. It's always great to have people like in our comment session talking to us. So
Absolutely.
yeah, it's been a pleasure chatting with you all.
Same here.
Tune in next week for more microbiology content. Bye.
Bye-bye.